Good morning, everyone. Good afternoon or good evening, wherever you are. Thank you for joining this EMN series of webinars. And I'd like to thank you for registering. My name is Baljit Yubi. I'm a staff scientist at SciEx in California. I'm going to be hosting this webinar on behalf of the Early Career Members Network of the Metabolomics Society. This is actually the 9th webinar of our series and is brought to you by the EMN, the Early Career Members Network Committee. Currently the webinars are free for you to attend by the courtesy of the Metabolomics Society, and I'd like to inform you all that all of the material that we're going to be recording will be uploaded to our website, allowing you to access the live content whenever you wish. Next slide, please. Please do connect with the Metabolomics Society on Facebook, on Twitter, and on LinkedIn, as can be seen on your screen. I'd also like you to request feedback about the Early Career Members Network. You can share any ideas and suggestions using the email address that's on your screen. Next slide, please. So before introducing our speaker, I'd like to introduce you to the new Metabolomics Forum. It's a platform where you can talk and discuss any subjects that are related to metabolomics, the Metabolomics Society, whether it's hardware, software, databases, etc. And if you've got any questions related to metabolomics, you can actually post them on the forum and get answers from other users or experts in the field. So please do, I highly encourage you to visit the forum, create your own profile, and use the platform to talk and discuss anything about metabolomics. The link is displayed on the screen and is also on our website. Next slide, please. So today's expert speaker is Associate Professor Peter Meikle, an NHMRC Senior Research Fellow. He's the head of the Metabolism Program and of the Metabolomics Laboratory at Baker IDI Heart and Diabetes Institute in Melbourne in Australia. He's also the Editor-in-Chief of Metabolites and holds affiliate positions at the University of Melbourne and Monash University. The Metabolomics Laboratory has a focus on dyslipidemia and altered lipid metabolism associated with obesity, diabetes, and cardiovascular disease, and its relationship to the pathogenesis of these different disease states. The laboratory has developed a targeted lipidomics platform that's able to quantify over 500 lipid species in 15 minutes using liquid chromatography coupled to electrospray ionization tandem mass spectrometry. And before I pass over to Peter, what I'd like to make you all aware of is there is a questions box on the control panel on the right-hand side of the screen, and if you drop the plus button, you can ask any questions during the talk live, and I'll put those questions to Peter at the end of his presentation. So thank you very much, and over to you, Peter.
Okay, thank you, Bal, for the introduction and the invitation to speak at this webinar. This is a new experience for me, as I expect it is for a lot of people. Thank you all to the listeners for logging on to this presentation. Now, as Bal said, I'm at Baker IDI Heart and Diabetes Institute, and of course the focus here is on research into type 2 diabetes and cardiovascular disease. And so what I thought I would do through this talk is firstly to talk a little bit about the lipidomic strategy that we've adopted here at Baker IDI and the types of studies that we're trying to do, because that really dictates the lipidomic platform that we've been developing to address those questions. And then I'll go through and describe the platform that we've developed and then go on to talk about a couple of examples where we're using that platform to look at some large cohort studies to investigate cardiovascular disease. Okay, so we're interested in this disease continuum that begins with obesity, progresses through to insulin resistance syndrome, such as including hypertension and general dyslipidemia, as well as type 2 diabetes. And of course, these are all risk factors for atherosclerosis and cardiovascular disease. And so there are a number of questions that we want to ask around these different stages of metabolic disease. And these include the question around biomarkers. So we're interested in identifying and developing new biomarkers to predict disease risk and to predict outcomes for these disease. We, of course, also want to understand the relationship between lipid metabolism and disease onset and progression. So it's really a question of pathogenesis in these disease states. And of course, these same biomarkers may be useful in assessing response of individuals to different treatments in these disease states. And finally, we have a particular interest in this question of stable versus unstable atherosclerosis. And this question of stable versus unstable disease really relates to the stability of the plaques that build up in the coronary arteries of patients, where unstable plaques are liable to rupture, and this results in a heart attack or stroke. And so this is a very important clinical outcome in terms of cardiovascular disease, and in fact the example that I'll talk about today relates to this. Okay, so how do we address these questions then that we have around this metabolic disease continuum? Well, the approach that we've taken at Baker IDI is really to go into biobanks or clinical trial samples where we can identify subsets or sub-cohorts of these patient samples that address different stages of the disease continuum. And then with those cohorts of patient samples, we can then apply different profiling strategies. And of course, mainly we've been working in this metabolomic and lipidomic area, but we're also looking now at some proteomic and genetic profiling as well. And in fact, the integration of the lipidomic with proteomic and genetic profiling, I think, is going to be very important in the future. So that's our strategy. And of course, the main sample that's available from those patient cohorts, or either from population studies or from clinical trials, is plasma. And so quite early on, we decided to develop a plasma lipidomic platform at Baker IDI. Now, of course, we know that plasma contains these lipoprotein particles, so LDL and HDL, so low and high density lipoproteins. And these are clinical measures that are already used for risk assessment in type 2 diabetes and cardiovascular disease, along with triglycerides and cholesterol. But of course, we also know that these lipoprotein particles are themselves very complex mixtures of many different types of lipids, many different classes and species of lipids, combined with different proteins. And so these lipids include things like ceramides and sphingolipids, which are important signaling molecules. The phospholipids make up the major component of the outer layer of these lipoprotein particles, and the di- and triacylglycerols and cholesterol esters make up the inner hydrophobic core in these lipoprotein particles. Then, of course, there's a range of modified lipids that might be oxidized or glycated that again may reflect disease states. There are lysolipids, fatty acids, and all of their metabolites. So many thousands of different lipids in humans, and in fact most of these will be present in plasma at some level. Of course, whether they're abundant enough to be detected with current methodology remains to be determined for many of them, but our estimates are certainly there's more than 1,000 lipid species that we could fairly readily detect with current methodology. And so our approach then has been to not necessarily measure all of these, but to get a representative profile of these relatively abundant species in human plasma. And our platform has been working towards that end over the last few years. And so we looked initially at, well, what are the major categories of lipids that we should be focusing on? These are the 8 categories listed in lipid maps. Not all of these are relevant to mammalian systems, and we've crossed out a couple. Prenol lipids, while they are present in mammalian systems, are not particularly abundant in circulation, and so we haven't focused on those. Fatty acids, which are quite important, again, the fatty acids themselves are not particularly amenable to liquid chromatography. Typically, these are measured by gas chromatography, and the eicosanoids and prostaglandins, so these are fatty acid metabolites, are very important in signaling processes, but again, a fairly low abundance in circulation. And so although we've looked at some of these, they don't, or they're not amenable to the very high throughput lipidomics that we're mostly doing. So where we've really focused is on the glycerophospholipids or phospholipids, and these are some of the major classes listed here, and these include the lyso species as well as alkyl and alkenol species or plasmalogens, which we have a particular interest in. The sphingolipids, which as I mentioned include ceramides and other signaling molecules, and of course the glycerolipids, so these are the di and triacyl glycerols as well as cholesterol and cholesterol esters and other derivatives. So these four categories are where we've put most of our effort. And the process that we've gone through to develop this platform is to start with a lipid extract from human plasma. We apply a number of precursor ion scans and neutral loss scans to that plasma extract as we elute from a liquid chromatography column. And this allows us to identify the major species within each of those classes of lipids that I mentioned earlier. Once we've identified those, we can then use the information that we've generated to establish what are called multiple reaction monitoring experiments, and I'll talk more about that a little bit later. But this is the general workflow to develop the methodology. And for those that are perhaps less familiar with mass spectrometry, most of the work we're doing is on triple quadrupole instruments, and the precursor ion scan that I mentioned essentially involves using the first quadrupole to scan through the mass to charge ratio, selectively allowing different lipids to enter the second quadrupole where we fragment those lipids. We use the third quadrupole to filter on a particular product ion that goes through to the detector. And so what this type of experiment does is it identifies all of the lipids in our original mixture that fragment to give a particular product ion. And since lipids within a single class typically fragment in the same manner, we can use this type of approach to identify all the lipids within a given class. And an example of that is shown here, where we're looking at, in this case, a ceramide, C16 ceramide. It fragments in a particular and predictable way within the mass spectrometer, so it loses a couple of water molecules and this acyl group here to produce a sphingosine backbone with a characteristic mass of 264, or mass to charge of 264. So if we perform a precursor ion scan for this mass to charge 264 across our chromatographic run, we see a number of peaks coming up that correspond to ceramides, and if we extract out the spectra corresponding to the signals that we've acquired, we can start to identify specific ceramide species that are abundant in the human plasma that we've started with. And so there's a couple shown here, as well as our internal standard, which is a ceramide with an odd carbon chain backbone. Okay, so with that approach we can step through the different classes of lipids that we're interested in, and we can identify the major lipid species within each of these classes, and this is just showing a partial list of the lipids that we can identify with this precursor ion scan of 264. And you'll see here we've got 6 species of ceramide and our internal standard that we've added into this. And for each lipid class we typically add in a single internal standard species, and some of these are deuterated standards and some odd chain fatty acid standards that are either not present or very low abundance in human plasma. Okay, so with that information of what the major species are, we then use this multiple reaction monitoring experiment similar to the precursor ion scan, but in these experiments we set Q1 to only allow through a single mass to charge ratio, again fragmenting in Q2. Q3 allows only a single fragment through. And so we have this effective double mass filtering in this type of experiment. And again, so it gives us a very specific measure of a particular lipid. The advantages of this, in our hands at least, you know, for triple quadrupole instruments is you get this increased specificity and you reduce the background noise by this double mass filtering. Using this type of approach, you also can look for longer periods of time at individual lipids, and so you can improve sensitivity. And of course, we can couple together multiple analytes by just simply defining multiple MRM transitions, or these Q1 and Q3 ion pairs, and we can do that then in a single experiment. So typically in these experiments, the dwell times can be only a few milliseconds in length, and so we can easily fit maybe 50 to 100 analytes in a single cycle time, which might be elution time for these analytes. And then if we schedule those experiments, we can quantify perhaps 500 to 1,000 analytes in a single chromatographic run. The downside, of course, is you do need to do all this work up front to characterize the lipids that you want to analyze. Okay, so the platforms that we're using at Baker are an older SciEx 4000 Q-trap we've had for about 8 years, but still working very well for this type of work. And then more recently, we've been working on a 6490, again, both triple quadrupole instruments. And we couple these with LC systems, and we're using fairly short columns initially, at least, C18 columns, 1.8 micron diameter and 50 millimeters in length. Okay, and so we've gone through this process, and initially the first iteration of our profiling platform contained these lipid classes, and these are the number of species that we identified in each of these lipid classes. So we had about 350 lipids. You can see phosphatidylcholines and ethanolamines made up almost half of the lipid species we're looking at. And in fact, these break down into a number of subclasses, and these are the ether lipids and the plasmalogens that I mentioned earlier that are very important, I think, in this sort of metabolic syndrome associated diseases of diabetes and cardiovascular disease. Okay, so the other aspect to the development side of this was really to work up a lipid extraction process that would allow us to work through hundreds to thousands of samples in a reasonable time frame. And so we spent quite a bit of time working up this process of starting with very small samples of plasma, because again a lot of these cohorts don't have a lot of samples, so we use typically 10 microliters of plasma. We add in our internal standards. We do a single phase butanol methanol extraction with 100 microliters of a 1-to-1 mix of butanol methanol. We sonicate that for an hour, we centrifuge, take the supernatant, and that's injected directly into the LC mass spec system. So this is really quite simple. We can extract maybe 500 samples in a day with that type of approach, and then these go into our system that I've just described. Okay, and the output then from that type of analysis is shown here. We get this series of chromatograms over, in this case, a 10-minute LC run. Early on we have the lyso phospholipids eluting, then we've got sphingolipids and phospholipids. Then we have diacylglycerols and the more hydrophobic triacylglycerols and cholesterol esters elute towards the end of these analysis runs. Initially we were working just with linear gradients using a mixture of methanol, THF and water, and that worked fine, and we went through a lot of samples with this type of approach. Of course, there are limitations to any approach, and some of the limitations to the approach I've just described is that, firstly, we're only working in positive ion mode. Because we're trying to analyze many hundreds or many thousands of samples, we don't want to have to analyse each sample twice, and certainly the instruments we've got are not amenable to the very rapid switching back and forth between positive and negative ion mode. So we've really set up everything that we're looking at in positive ion mode only. And so there are some lipids that perhaps we can't include in this. This type of approach, because we have only one internal standard per lipid class, the quantification that we obtain is a relative quantification only, not an exact quantification. We're not setting up calibration curves for every one of our 350 lipids. So we recognise that there's a limitation in the accuracy that we're able to get for each individual lipid species. What we focus on, though, is that we have very precise measurements, and so that within an analytical run, all of the samples are comparable to each other, so we can identify lipids that are increased or decreased in a particular disease state. In some instances, we do apply correction factors to the lipids where we've been able to determine, either using standards or other means, what the response factors for given lipid species are, but for most of them we don't apply correction factors. And of course there are limitations in terms of the resolution that we can obtain between isobaric and isomeric species. Again, this is particularly important where these species have the same fragmentation, and I'll talk about that a bit more in a minute. And so because of these limitations, it's not always possible to get the exact structure of a lipid species using this approach. And so here's an example of isobaric and isomeric species that we're including in our analysis. So we've got a plasmalogen, and the structure shown here, where we have this vinyl ether linkage on the SN1 position of a phosphatidylglycerol, and we've got a arachidonic acid on the SN2 position. So this gives us a nice signal, and this is clearly distinguished from the isomeric species, which is this PCO36-5, which has a very similar structure but simply has a double bond further down this acyl chain, and so has an ether linkage here rather than a vinyl ether linkage. But because we're able to separate these chromatographically, we can get quantification on both of these individually. And similarly, we have odd chain fatty acids incorporated into some phosphatidylcholine, which have the same mass, not the same exact mass, but in terms of the resolution of a triple quadrupole instrument, we can't distinguish these based on mass. So we need to rely on the chromatography to be able to distinguish these. But in this case, these work quite well, even on the relatively short chromatography that we run on our profiling platform. What we can't do with these shorter chromatographic runs, though, is separate things like this PC species, or these couple of PC species that have a combination of 36 carbons and 3 double bonds. So that can either be a mixture of an 18-1 and an 18-2 fatty acid, or a 16-0 and a 23 fatty acid. So both have exactly the same mass, both fragment in the same way, and so without resolution in the chromatographic setting, we can't distinguish these, and so we report them initially as a PC36-3 and recognise that it's a mixture. However, what we've been doing just over the last 12 months or so is changing our chromatography. So we've increased the length of the column to a 100mm column. We've done a lot of work on the gradients that we're using, and so we've managed to stretch out a lot of these, the lipids and the area that they're eluting in right across this profile. And so typically now our chromatography runs for between 15 and 20 minutes, depending on the instrument. But what it allows us to do is go from this type of resolution to this type of resolution, where we can now differentiate these two different phosphatidylcholine species, and of course there are many others that fall into this type of class. We also, with this improved chromatography, start to get separation of the lysophospholipid into two distinct species for each lysophospholipid, and this is all of the lysophospholipids we're measuring. And what that corresponds to is whether the acyl chain on the lysophospholipid is in the SN1 position shown here, or whether it's attached at the 2 position. Now these are fairly subtle differences in terms of structure, but the metabolism and the biology behind the production of either an SM1 or SM2 lysophospholipid is quite different. So it becomes quite important in certain situations to be able to differentiate these two species. Okay, and so without going through all of the highly complex chromatograms and characterization, a couple of people in my lab over the last year have done a lot of work to resolve these and characterize these different species, and most of this work has revolved around characterization of the phospholipids as well as some sphingolipid species. And this is really based on going into detailed analysis of fragmentation in positive and negative mode. We use things like lithium adducts to give fatty acid specific fragmentation, particularly of those phosphatidylcholines. Hydrolysis to selectively hydrolyze plasmalogens, and so identify where you have complex mixtures, which species are plasmalogens and which are either alkyl or diacyl species. So using those types of approaches, we've then expanded our profiling from the 350 species we had initially to just around 600 now. We've added a couple of new classes. We're looking at sulfatides and some additional ganglioside species as well as acyl carnitines. But mostly we've just been expanding and getting more detail of all of the phospholipid classes. Again, phosphatidylcholines and ethanolamines make up a large proportion of these. And again, we can expand those out into these different subclasses. And of course, we should also remember that while the lipids exist within classes, the classes themselves exist within metabolic pathways, and this is just showing a partial metabolic map. And highlighted here is the sort of coverage we can get with our lipid profiling strategy. Okay, so let me move on now to talk about how we've used this platform then to look at prediction of cardiovascular events. And the area that we're interested in is in secondary prevention. So secondary prevention refers to treatment of patients who have established cardiovascular disease, and this consumes a lot of resources, and that's due in part to our inability or limited ability to identify who's at high risk and who's at low risk in this group of patients. And so we wanted to ask whether lipids could help in this risk prediction in this secondary prevention group of patients. The study that we had available to us, or the cohort that we had, was from the lipid study. So this was a long-term intervention with pravastatin. So it was one of the early statin clinical trials conducted in the 1990s, but we still had samples available from this study. It was a double-blind placebo-controlled study, about 9,000 participants. Everyone had a history of either acute myocardial infarct or unstable angina, so heart disease. They had cholesterol in this relatively high range, 4 to 7, and they were randomized into either receiving a pravastatin treatment or a placebo. And the outcome from these studies was really to look at cardiovascular events over a 5-year period. And so they looked at everyone who had a myocardial infarct or a heart attack, a stroke, or who died from cardiovascular disease-related causes. And so we've combined all these into cardiovascular events, but we also look at cardiovascular disease-related death as a separate outcome. So we didn't have all 9,000 samples from these individuals, but we had about 5,000 participants where we had both a baseline sample and a 1-year follow-up sample. We had another 1,000 samples just from baseline and about 800 where we only had the follow-up sample. So in all there was about 12,000 samples. And when we looked at the event rate in these, we can see we had about 700 cases of cardiovascular death, close to that 650 heart attacks, and about 300 stroke. So we had a lot of outcomes and a lot of power then to look for associations between lipids and these clinical outcomes. Now the important point about clinical trials is that they have very good phenotyping of all the participants, and so we had all of this clinical data on all of the participants, and you can see that a lot of these characteristics also associate with the cardiovascular events, and so are in themselves risk factors for future cardiovascular events in this cohort. And of course, what we were interested in is whether we could identify lipids that could improve upon all of these clinical risk factors for future cardiovascular events. And so these were all used in our subsequent analysis of the data. Our hypotheses were that the baseline plasma lipids in this cohort would be associated with incident cardiovascular events, and that this would be independent of all those traditional risk factors. And so our aims were then really to perform the lipidomic profiling on our 12,000 samples that we had, identify species that were associated with future cardiovascular events, and then to use those lipids to develop multivariate models to predict future cardiovascular events. So using our high-throughput lipid extraction method and then high-throughput targeted lipidomic method, we processed all of those 12,000 samples, or just under 12,000. At this stage, we were measuring around 350 lipids, and in a 10-minute run. So this was about 4 months continuous run time on the mass spectrometer. We ended up with a very large data set. We had to align this and normalize the different batches using median centering to get the data all aligned. But we ended up with this data set of about 4 million lipid measurements over those 12,000 participants. And then we could start to ask questions, well, which lipids associate with our different outcomes? And as I mentioned earlier, the outcomes that we're interested in are either all cardiovascular events combined, so heart attack, stroke, and CVD death, or simply looking at CVD death on its own. And to do this, we used a Cox regression analysis. So we firstly log transformed the data, normalized it to the standard deviation so that different lipids, we could interpret different lipids in the same manner. We adjusted for all of the covariates that we had identified earlier. So we actually had 22 different covariates that were clinical or traditional risk factors for cardiovascular disease. And then we calculated hazard ratios and 95% confidence intervals. We corrected all of our analyses for multiple comparison because we're analysing many hundreds of different lipid species. Okay, so of course it's difficult to show you all of the data that came out of those analyses, so I'm just going to take you through a few specific examples. So one of the first analyses we do is actually not look at the individual species but look at the lipid classes. So we sum the individual species within each class to get a class measure, and then we do the analysis just on lipid classes. And so when we do that, we can see that there are two classes that show an association with cardiovascular death in this case. And we see this positive association indicated by a hazard ratio greater than 1. So this wasn't particularly exciting or productive, but in fact, when we go in and look at the individual lipid species within each of these classes, we see that there are a lot more individual species that are showing up, but whereas the class itself doesn't show up as a significant association with future events. So we had 55 lipid species associated with cardiovascular events and 95 associated with cardiovascular death. So if I just take you through a couple of examples of what we see and also why we're not seeing these associations with the lipid classes. So for example, here we've got a series of, firstly, dihydroceramides. We measure 6 species in this, and we see that only 2 of the 6 species show this positive association with future cardiovascular death, but that the species that contain the longer chain fatty acids, so 20, 22, 24, and so on, don't show this association. Then when we look through at the ceramide species and the monohexacyl ceramide species, we see the same pattern. So these first species that contain 16-0 and 18-0 show this association, but the longer chain species don't. And so to try and put this back into a biological context, we can go back to our lipid metabolic pathway. And what we see is that this pathway of sphingolipid synthesis starts with serine palmitate, we have the synthesis of dihydroceramide, and this is metabolized through this pathway to ceramide, then to mono, di, and trihexacylceramide. And in all of these lipid classes, we see this common feature of the 16-0 and 18-0 fatty acid species show this positive association, but there's no association with the longer chain lengths. And what we see if we look up more about this biosynthetic pathway is that there's a series of enzymes called ceramide synthases that show specificity for different fatty acid chain lengths. And so what appears to be happening in this situation is that we've got a dysregulation or an upregulation of some of these ceramide synthases in patients that are at high risk for a heart attack or stroke, such that we see only these shorter chain species of ceramide and the subsequent metabolic products being increased in those patients. So again, this is starting to point then to potential sites for therapeutic intervention. So we could potentially target these individual isoforms of ceramide synthase to try and reduce risk for future cardiovascular events. And in fact, there's a number of groups working on this at the moment. Okay, another story that came out of this analysis was looking firstly at phosphatidylcholines here, and again, in this case, we see species that contain arachidonic acid, or 24, tend to show a negative association in phosphatidylcholines, whereas these species containing shorter chain, monounsaturated and saturated species tend to show no association. Whereas in phosphatidylethanolamine, we see an opposite effect where the shorter chain species are showing a positive association with future cardiovascular death in this case, whereas the arachidonic acid, while still positive, is not significantly associated with future outcomes. But we are seeing generally a shift of all of these species to a more positive association. Where we looked at other lipid classes, other phospholipids as well as cholesterol esters, di and triglycerides, we see the same effect. That is, species containing arachidonic acid tend to show a negative association. Species containing saturated and monounsaturated fatty acids tend to show a positive association. So again, if we come back to our metabolic map, we can see that there's this upregulation of this phosphatidylethanolamine pathway in patients that are at high risk. And this includes an upregulation, although not significant, of this arachidonic acid containing species of phosphatidylethanolamine. And this comes at a price of a decrease in the level of arachidonic acid contained in a number of other lipid classes. And so we're seeing this, again, dysregulation of, in this case, arachidonic acid metabolism through different pathways. And since arachidonic acid is the precursor to the eicosanoids and other pro-inflammatory molecules, then again, this may be mechanistically important in terms of inflammation in these patients at high risk of cardiovascular disease. Okay, so that's some of the biology that can come out of these types of studies. But of course, we also want to see whether lipids can predict who will be at greatest risk of future cardiovascular events. And to do that, we want to make multivariate models. So is there a combination of lipids that are better than any individual lipid? Of course, we don't want to use 350 lipids in a single model. It would be difficult to translate that into any clinical application. So what we want to try and do is firstly create a model with the minimum number of lipids, but a model that has the maximum power to predict future risk of cardiovascular events. And so the first step in this process is to minimize the number of collinear lipids or highly correlated lipids, and we use this process of correlation minimization to do that. And we reduce from 350 down to 140 lipids or so by removing things that are lipids that are highly correlated. And of course, we also want to create a model that we can demonstrate improves upon all of the clinical risk factors for cardiovascular disease. So we initially create a model using all of our covariates, so these are the traditional or clinical risk factors, again using Cox regression. And then to this base model, we sequentially add our lipid species of interest. And we select those lipid species based on a Aki-Aki information criteria. That's the goodness of fit of the model in terms of stratification of our events and non-events. All of this modeling is done within a cross-validated framework of 5-fold cross-validation, and we've repeated a couple of hundred times. And from this, these 1,000 iterations, then we can identify which lipids are most often included in these models, and so we can rank the lipids in terms of their importance in the development of this model. And so with those 20 top-ranked lipids, we then go back and create a new series of models, again by adding lipids to our base model, again within a cross-validated framework. But here we focus on measuring the C statistics, so the area under the curve, and the net reclassification index, which is another measure of model performance, and the associated 95% confidence. Again, back to our base model. So this is some of the outcome from those analyses. We can see here what we're looking at is the average C statistic from our models, where we start with the base model, shown here as 0, and then as we add additional lipids into that base model, we see that the C statistic improves up until about maybe 8 to 10 lipids are added, and then it tends to level off, predicting cardiovascular events or in fact cardiovascular death. We get a similar profile. We can translate that then into numbers, and so here we're looking at firstly our base model, and then our base model plus the 8 lipids which we identified as being the sort of optimum in these particular analyses. And so we can see that we get about a 2% increase in the C statistic to predict cardiovascular events, and this correlates to an 8% improvement in the categorical NRI and about a 30% improvement in the continuous NRI. And then we did the same analysis looking at cardiovascular death, where we see a slightly better performance, about a nearly 4% improvement in the C statistic, 10% improvement in the categorical NRI, and about a 40% continuous net reclassification index in these. Okay, and this is just showing you the lipids that were included in those models. You might see your favourite lipid here, or maybe you won't, but the models for cardiovascular events and cardiovascular death were had 4 common lipids, but also a number of different lipids. You know, again indicating that you really need to focus the model on a particular outcome. And in fact, we've done a number of other studies to look at differentiating myocardial infarct from stroke, and again we see that there are different lipids required for different cardiovascular events. Okay, so in the last couple of minutes, let me just talk about validation of these findings. Of course, with all of these biomarker discovery and modelling type of studies, it's quite important that we can validate findings on an independent cohort. And so we had another cohort from the advanced study, and that was this study looking at diabetes and the effect of either blood pressure control or intensive glucose control in diabetes. So again, another double-blind placebo-controlled study, 11,000 participants, all type 2 diabetic patients. These were the interventions that we used in this 2x2 design. The outcomes were the same, so primary outcome was 5-year follow-up for cardiovascular events, including MI, stroke, and cardiovascular death. Again, we didn't analyse all 11,000 participants, but we had about just over 3,000 participants selected at random, which gave us about 330 cases, so MI, stroke, and CVD death. And then we enriched this sub-cohort with all the other available cases that we had from the rest of the trial, which were a further 360 cardiovascular events. So that gave us this final cohort of about 3,800 participants and about 700 cases. So we did all of the same analyses that I described for the lipid trial. And what I'm showing here then is looking at all of the lipids that were significantly associated with cardiovascular events or cardiovascular death in the lipid trial, shown on the x-axis or the y-axis. And we plotted the hazard ratios in the lipid trial with the hazard ratios that we see in the advanced trial. And so we see then there's a group of lipids here that are positive in both the lipid and the advanced trial, and similarly a group of lipids that are negative in both the lipid and the advanced trial. Of course, you can see some that are either positive or negative in the lipid trial but are not significantly associated in the advanced trial. And this is perhaps to be expected as the advanced group, although it's a high-risk cardiovascular disease group, it is a type 2 diabetes group, and we know that there's a severe dyslipidemia associated with type 2 diabetes itself, and so we're trying to see the lipids associated with cardiovascular disease on top of this underlying dyslipidemia associated with diabetes. Nonetheless, these results were quite encouraging, and we went on to perform the same modelling that I described for the lipid study. That is, can we predict cardiovascular events using the same 8 lipids that we identified in the lipid study, but now applying them on this advanced cohort? And when we do that analysis and we look at what the improvement in the C statistic, we see a very similar improvement for cardiovascular events. The categorical NRI is not quite as high, but still significantly improved. And the continuous NRI is about the same as we observed in the lipid study. And when we look at cardiovascular events, cardiovascular related death, the outcomes are again very similar to what we saw in the lipid study. An improvement in the C statistic, 10% improvement in the categorical NRI, and about 40% improvement in the continuous NRI. Okay, so what we've been able to show, I think, then is that lipid studies are associated with future cardiovascular events in secondary prevention. This is independent of all of the traditional risk factors and clinical factors that are currently used. And we can also see that we're starting to see some biological insight as to lipid metabolic pathways that may be relevant in this disease. We can use those plasma lipids in a relatively small group of them to predict cardiovascular events. And again, we've been able to validate that in an independent cohort. So thank you for listening. I'd just like to acknowledge the team here at Baker IDI, our collaborators at Baker IDI, particularly Bronwyn Kingwell and Paul, our collaborators at the Clinical Trials Centre in Sydney, John Symes and his team, as well as the George Institute, where these are the institutes that conducted these large-scale studies, and of course we work closely with colleagues at Melbourne University as well. So I'll stop there. I do have one final comment. I'd just like to make a quick plug for the 3rd Australian Lipid Meeting, which will be held in Melbourne, in fact at Baker IDI on the 21st and 22nd of November this year. You can visit the website to find out more about it, but we would love to have some overseas visitors to our meeting. The first 2 meetings have been very successful, and we're sure this one will as well. So I'll stop there and happy to take questions if there are any. Thank you.
Thanks, Peter. Thank you so much for such a wonderful and an insightful presentation. I wonder if you can just click the slide to the next slide for me.
Yes.
And so before we begin the question session, what I wanted to announce was the call for applications for the EMN committee, and we're looking for at least 5 additional early career scientists. The eligibility is listed on your screen, and you can actually send your applications to the email address that's there, and the deadline is actually the 15th of August. And if you have any questions, do shoot us an email via this email address. Okay, so I actually have quite a lot of questions for you, Peter, and I've been trying to sort through them for...
Excellent.
For anybody who hasn't had a chance to ask any questions on the screen. Okay, so first question. was actually right off the back of probably your first or your second slide around not just the metabolomics data, but also the proteomic data and the genomics data that you're collecting. Have you started to combine those omics data sets?
One of the largest studies that we've done has been on the San Antonio Family Heart Study. This is with a collaborator in Texas in the States, John Blangero. So again, this is a population study. John's done some large SNP arrays on these, about 1,200 individuals. This is a large family study. And we've done the lipidomics on these, and so we have started to combine this, about 1 million SNPs with the lipidomic data to identify, firstly, associations between lipids and polymorphisms in the genome, and then relating both of those back to clinical outcomes in terms of diabetes and cardiovascular disease. So this is looking very promising, and I think the family cohort that John's put together in this is very powerful in this setting. There's still a long way to go. In fact, John's just in the process of finalising the whole genome sequence on this cohort, and that will give us a lot more power. But yes, certainly that's one aspect that we're looking at, and we've got a couple of other studies that are underway where we're still acquiring data to combine genomic and lipidomic studies. We've got I'm working with another group here at Baker IDI looking at a group of diverse mouse models that we've combined both lipidomic data as well as proteomic data, and so we've got our sort of 500 lipid measures and about 5,000 protein measures from the liver of these mice, and we're again just starting to put that data together. But certainly I think this is going to be a very productive path forward for lipidomics and metabolomics in general.
Sure, so look forward to present that data sometime in the future. How does, so this is the next question, how does the butanol, methanol extraction that you discussed compare to the traditional more BLIDIA extraction?
Yeah, look, we've done a lot of validation of that methodology, and that's published in Metabolites. I think it was last year or earlier this year. Essentially, the advantage of the butanol methanol over a Fulcher or BLIDIA is, firstly, it's a single phase extraction. In a Fulcher or BLIDIA, you run the risk of losing some of the polar lipids in the aqueous layer, or some lipids you also tend to lose at the interface between the aqueous and the organic phase, whereas we don't have that problem in a single phase. In terms of percentage recovery, we get all of the lipid classes that we've looked at, we're getting mostly above 95% recovery of lipid classes, and for all of them it's above 90% recovery. So the recovery is very high in this process, and a lot of it relies on giving that fairly long sonication that we use in this particular extraction process. But of course, because it's a single phase, then we're not removing very much of the salt, so you need to then combine it with liquid chromatography to remove the salt before the sample goes into the mass spec. These samples wouldn't be particularly suitable, I don't think, for direct infusion, for example.
Okay, next question, and actually there's two questions that are very similar, so I'll put the first one to you. You mentioned that you could actually differentiate between the SN1 and the SN2. The question here is that they actually interconvert in solution. Do you have a strategy for avoiding that? And how do you interpret the results?
Yeah, look, we're looking at that now. We've done a bit of work. There is a strategy to avoid that, and obviously this is probably, that SN1 and SN2 differentiation is probably less important in plasma, where I think that for the most part, they've probably reached an equilibrium. But if you're doing the same sort of analysis on cell extracts or on tissues from animals where you can collect them, you know, fresh and treat them a bit more carefully than the plasma samples, you can collect into acidic environments, and that that slows down the interconversion. And then when you extract, you can also extract into acid solutions to minimize the interconversion. And so we are able to see in cell extracts, for example, differential SN1 and SN2 expression under different conditions. And sorry, what was the second part of that question?
So the second part, I think it was more around clarity. So I think you showed a slide where you could actually differentiate the SN1, SN2 of the lysophospholipid. And so the question was, what basis did you do that? I think that was based on the fact that you expanded your chromatography, or can you clarify a little bit more how you were able to do that?
Really, the differentiation is really just based around the chromatography. If you you know, again, with the longer columns and the you know, very small particle size, you can separate out that SN1 and SN2 fairly easily. But you know, I take the point that interconversion, particularly in plasma, is probably an issue.
Okay. There's a question here from somebody who's getting started in lipidomics. I'm new to lipids. Is it possible to measure lipids in serum? And if so, what's the expected difference between the quantity, quality versus plasma?
Yeah, and we do serum as well, although I guess if we have a choice, we typically go for plasma. There's not a big difference between serum and plasma. Of course, you know, all the same lipids are present. The relative amounts will vary a little bit, but it's really only a few % difference between plasma and serum. The issue is really, it's fine to use serum or plasma, but not a combination of both, because then obviously you're adding extra noise into the system. So if you've got a cohort that's all on serum, then that's fine, or a cohort that's all plasma, that's fine as well.
Okay, and last question, I think, before we sort of close up. The pathway mapping that you showed
Yeah, I'm not hearing you, Val.
In one of your slides. Hello? Can you hear me?
Yeah, I can hear you now. Sorry, I missed that question though.
Okay, sorry about that. The question was around the pathway mapping that you showed in one of your slides. Is there a more automated way to map lipids to pathways rather than doing it very manually?
Yeah, gee, I wish there was, but not that I'm aware of. You know, there are some good databases now to search for lipids if you're trying to identify new lipids based on mass or even fragmentation. But in terms of pathway analysis, I'm not aware that there's any truly automated process for that. You know, we've just really put those maps together from, you know, information in the KEGG databases and things like that. And you can certainly pull out the major metabolic pathways out of the KEGG database, but again, they tend to be overly complex, and when you're trying to display just a few lipid classes or individual pathways. So no, unfortunately, there's no easy way to do that.
Okay, so thanks again, Peter. Thanks for answering the questions. Sorry that we couldn't get to all questions. I just want to end by thanking Peter for his time today. I want to make a little plug for next year's Metabolomics Society meeting, which is actually going to be held in Brisbane. So look forward to seeing many of you there, and hope that you can actually get on the website and onto the forum and start engaging with us. And I'd like to say thank you and end this webinar. Thank you very much.
Okay, thanks, Belle. It's been a pleasure.